Today we are going to discuss about the internal structure of anther. So you have already come to know in our previous lecture that anther is of basically two types. One is called monothecous, which contains only a single lobe and it looks like this. And if you take a transverse section of an anther, it will look like this from upper position. And another type is called bithecous, and bithecous anther has two lobe structure, that means bithecous, and it looks like this. And if you take a transverse section of this type of anther, it will look like this. Now we have to know about the internal structure of an anther. You see, the internal structure of an anther looks like this. As I have seen that bithecous anther has two lobes, that means 4 microsporangium is there, and these are the 2 lobes, lobes number 1 and lobes number 2. Suppose lobe number 2. So here we will see what is the internal structure of an anther. You see, the anther has a 4-layer wall. 4-layer wall, and you see this is the relative position of microsporangium. We'll try to draw the wall which is of 4 layers. You see, this is the first layer. If we draw the cells of first layer, it will look like this. The first layer is of a single cell. That means this cell remains in this manner. Now you see, this layer is known as epidermis. The first layer is known as epidermis. And you see, the second layer is of also a single cell. This will look like this. These cells will look like exactly these cells. You see, this second layer is known as endothecium. And the third layer, that means which remains just beneath the endothecium, is of multi-layered cell. That means here cells may be 3 or 4 layered. This layer is known as middle layer. And just beneath the middle layer, one more layer is there, host cells are like pyramid shaped. To some extent it looks like pyramid, not exactly pyramid but to some extent. So this layer is known as tapetum. This is the most important layer of anther wall. You see, we have seen 4 different layers which has composed the anther wall. So this is the complete anther wall. Anther wall. Anther wall. Now you see inside, this is called microsporangium. Microsporangium. Microsporangium, inside which pollen grains develops later on. Now you see inside microsporangium some small cells are there and they remain densely crowded in this way. And these cells are known as sporogenous cell. Sporogenous cells, and the sporogenous cells have the capacity or capable to produce microspores. Capable to produce microspores. Now, till now we have come to know that anther looks like, that means if you take a TS section of an anther, it will look like this, inside which you will get 4 microsporangium, 1, 2, 3, 4 microsporangium, and before getting, that means if you move on to the inside from out, that means if you move on to inside from outside in this direction, you will get a anther wall, and the anther wall is composed of 4 different layers. The first one is called epidermis, the second one is called endothecium, third one is called middle layer, and the remaining, that means the last one is called tapetum, which remain attached to the microsporangium. And you have come to know this structure. That means this is almost a circular structure, and this structure is known as microsporangium, inside which some sort of cells are there, and those cells are termed as microsporogenous cells, and microsporogenous cells are capable of producing microspores. That means in later stages of development of anther, some sort of spores will be created inside microsporangiums by the cells of sporogenous cells, and these cells will be later on termed as microspores. Now you see, we have to know the function of these 4 layers. That means the first 3 layers usually helps in the protection of the microsporangium or the entire anther, and the tapetum, which is the layer that remains attached to the microsporangiums, provide nourishment. It provides nourishment to the microsporangium. so that inside microspores and other different activities can be taking place. And these three layers, that means epidermis, endodermis, so endothecium and middle layer basically provide protection and it provides nourishment to the sporogenous cells. So this is the scenario of an anther. If you take TS of the anther, you will be getting this sort of scenario and structures. So thank you.